And I'm pleased to invite Dr. Uday Saxena for his talk on omics technology in target selection and drug discovery process. I would like to take the pleasure of introducing him. Dr. Uday Saxena is currently a co-founder of the startup biotech company ReaGene Innovations. He has held executive and leadership positions at Parke-Davisin Ann Arbor, AtheroGenics in Atlanta as VP Drug Discovery, Dr.Reddy's Laboratory as Chief Scientific Officer, US and India, and Kareus Therapeutics CEO. He has a Ph.D. in biochemistry from Memorial University and post-doctoral training at Columbia University. He was associated with the team at Parke-Davis, Pfizer, that discovered Lipitor and atorvastatin, the largest selling peak sales of $17 billion drug in the pharma business. He led teams that brought several drug candidates from an idea into the clinic. And we are glad to hear from you, sir. Over to you.
Thank you very much, Sonalika, and thank you, Mohit. It's a pleasure to be here. Let me now try to play the slides myself. I have a 50-50 track record. Sometimes I'm able to do it, sometimes I'm not. I still haven't quite figured that out, but let me try it. Can you guys see my screen? Oh no, I have to do a screen share, is it?
No, sir. Yes.
Okay. How do I? Okay, one second. You'll have to let me do it. It's not letting me do it right now. So it says one participant.
So can you try now?
Okay. Okay. Can you see it now?
No, sir, we can't see it. If you click on the share screen icon, it will ask you this window or a specific slide deck that you would like to share. You can click on it.
Voila, can you see it now?
Yes, it's coming up.
Okay,
Yes, sir,
thank you. You know, you learn something new every day, so it's always a good thing. Let me now play. All right, so now you can see it, right?
Yes, sir.
Okay, thank you. Yeah, once again, thank you, Pine Bio, for a very kind invitation and introduction. As Mohit said, I'm going to tell you about omics technologies and specifically their applications in target selection and really all of the drug discovery process. So I will speak for about 25 minutes or so, and if there are questions, I don't know what the format is, but if there are questions, I'll be happy to take that. So briefly, I'll talk about, I think I should introduce drug discovery to this audience because I'm not so sure what your backgrounds are. So I think I'll go ahead and do a brief introduction on what it takes to discover a new medicine. Then I will plug into that the role of omics, and clearly omics have a huge role in discovery of new medicines or drugs. And specifically, I want to focus on proteomics and its role in drug discovery. Right off the bat, I can tell you that 90% of the drugs that we have today have a protein target. In other words, if we did not have proteins, we would not have had a drug. So that's how important proteins are in drug discovery. And as a result of that, proteomics has certainly assumed a very important role in discovery of new medicines. So this is to those of you who may not be aware, but discovery of a new drug is a very long process. And when I say very long, it takes anywhere from 12 to 15 years. The reason is as follows. If you look at the bottom, you will see that in the purple color starting, there's 3 phases to it. The first phase is what we call in vitro and in vivo testing. In that, basically, you start with suppose you want to go after Alzheimer's. What you have to do is figure out what is your target. So for example, in Alzheimer's, there is something called tau. Tau is a protein that gets deposited in the brain, as a result of which it's neurotoxic, and then that causes Alzheimer's. So the target for Alzheimer's drug discovery could be the tau protein. And there's a couple of approaches. Either you can block its synthesis or you can block its function. Either way, you can approach it. So that's how it starts. It starts with an idea of what target you want to go after. And then once you do that, as I will tell you in the next slide, there's some additional things you have to do, but from there, it moves on to early discovery, in which the chemists start to make drugs that might actually block the function of tau, whichever way you want. And then you have to select the best compound that becomes what's called a lead drug or a lead compound. And then you have to move into preclinical testing. Up until now, what I just talked about is all in vitro. And then as we move into preclinical, it's mostly in vivo work, starting with demonstrating that the drug you have chosen actually works in an animal model of Alzheimer's. And there's many animal models of Alzheimer's that you can do. And if it looks good, then you have to move into toxicity testing. Remember, a drug should not only just be effective, but it also be safe. In fact, I would say more than 50 to 60% of the drugs fail, not because they are not active or efficacious, but they're also toxic. So you want a drug that is safe, which is not toxic, as well as efficacious. Once you find that gem, you then move it into clinical development. This is in humans. You have 3 phases, phase 1, phase 2, phase 3. Phase 1 is typically in healthy volunteers, and then phase 2 in a small population of Alzheimer's. Phase 3 is in a very large population of Alzheimer's patients. And oftentimes you also have to compare it with what else is out there in the market. So all of this takes about the preclinical phase. Up to the preclinical phase, it might take about 5 to 6 years. And the rest of it really is clinical development, which means all the clinical trials. And something like Alzheimer's, which actually progresses very slowly over a period of years, it might take about 5 to 10 years to do the clinical development part. But if something like COVID-19, as we'll talk about it, is an infection, and that could, you could develop a drug there much faster than Alzheimer's. Once you successfully demonstrate that the drug is safe in humans as well as is efficacious for the disease, then you have to submit what's called a new drug application to the FDA, US FDA. They'll take about a year to review all of the data, and this is thousands of pages of data that you have to submit. And eventually, then you get approval. So it's about 10 to 15 years. It's generally $500 million, and the success rate is very, very low. You might start with 10,000 drugs that you look at, but only one is successful. So the probability of success is extremely low. So it's as you can see, it's a very complicated process, time-consuming, high risk, and takes a lot of money as well. So this is not for somebody who is weak-hearted. So drug discovery, now you know why new drugs, when they come on the market, might sound expensive. And the reason for that is so much of effort and money has to go in by the company to produce a new drug. Right? So this is a basic format of how new drugs are discovered. You need to know this before I tell you about how proteomics are important in this process. So the number one step that you have to have, as we already talked about in drug discovery and development, is you have to select a target. And I gave the example, you'll see in the green box. It's a strategic research decision you have to make. So the target could be tau. It could have been, amyloid also, or there are multiple targets in the Alzheimer's area you can go after. So you have to actually choose one. And then you have to validate it. So what validation actually means is if you manipulate the expression of the target protein, and you have to demonstrate its relationship with the disease. So for example, tau, if it goes up, it causes Alzheimer's. So you might take an animal model, overexpress tau protein in the brain, and demonstrate that you start to see Alzheimer's-like symptoms, right? So that's target validation. So before you put any money, time, and effort, you have to validate the target to the satisfaction of the company that you are working at. Once that is done, then you have to set up some assays, which are basically in vitro assays, to be able to say that if I add a drug, tau function is inhibited or not. That process, at that point, chemists come into play and you generate drug compounds that are called hits. Then you deal lead identification. Eventually, then you have to test in animals, in which you have to look at ADME PK, which is basically once you give the drug to the animal and eventually to humans, you want to know that it gets to the brain or not, right? Because that's where it has to act. So that process is called ADME PK. And then in clinical trials, you have to kind of repeat a lot of what you did in the preclinical animal studies. You have to kind of duplicate that there. So as you can see, it's all depends with the target that you select. If you did not select the right target, which is robustly involved with the disease process, you are going to fail. And the longer it takes for you to fail, the more expensive it gets. Clinical trials, for example, are about 80% of the cost. So off of $500 million, if you fail in clinical trials, then you probably lost about $400 million. It happens. Like I said, the odds of success are quite low. So the target selection, the protein that you want to go after, and its association with the disease has to be very robustly demonstrated and convincing. So just showing something in a petri dish and saying, I think this is going to work, is simply not enough, because you're setting yourself for a failure. So everything starts with selecting the right target. And usually, as I'll tell you, targets are 99% of the time they're proteins. So how do omics come into play here? Before we had the tools of the omics, like the genomics, transcriptomics, proteomics, and metabolomics, it was a little bit of a hit and miss. But now with these very sophisticated tools, you can actually understand the disease better. So to be able to select the right target, right, you should understand the disease process very well. Which step in Alzheimer's is tau coming into play? Does it come into play before the disease sets in or after the disease has already set in, right? Because once the disease has set in, then there is no point in going after tau. So you want to choose something that's upfront and upstream in the process. And it takes a lot of gene expression, proteomic analysis, and all of that. So not only in target selection, as I will tell you, does omics come into play, but also now our ability to understand disease processes with all these wonderful omics tools that are listed here has dramatically increased the likelihood of success of a drug. If you don't have an understanding of the disease, you will not be successful. It is as simple as that. So any tool that you can use to improve your likelihood of disease understanding, the better off you are there. So not just in drug discovery, in target selection, as I'll say, but this understanding the disease itself, all of the omics tools actually have a fundamental role to play. This is just reiterating what I said. I've broken it down more into the language of drug discovery. At the very bottom, you'll see, starting from lead discovery, target selection, and all of that. We just talked about every step of these have some form of omics being used. Sometimes it's genomics, sometimes it's transcriptomics, proteomics, or metabolomics. So at every step of drug discovery, even in clinical trials, when you are actually dosing patients with the potential drug, you can get their blood samples and do an array of omics technologies and try and better understand if the drug is working or not. So I think in summary for this slide, what I want to say is there is not one step in the drug discovery process in which we do not use some form of omics or not. So omics have really, really become very important in our quest for new medicines and drugs. So of all those that I talked about, you have the genomics, proteomics, and all of that. I think it's very safe to say that proteomics probably have the central role to play in that. That's simply because the target, the basis of designing a drug discovery program is selecting the right protein and understanding it in many different ways its function, its validation, what happens to it if a drug binds, all of that. So proteins, to my mind, are the perhaps, and proteomics thereby, are perhaps the most important tools that we have in drug discovery today. Again, I'm trying to tell you where proteomics plays a role. Obviously, target identification and validation. The target is a protein, so it's a no-brainer you'll need. Also in lead discovery and optimization. I'll tell you a little bit more later on about that. And molecular docking becomes very important as the scientists choose which is the right drug to move forward with, right? And then the in vitro and in vivo studies, even to understand the drug's toxicity and efficacy, you want to look at the drug might be affecting the protein that you want, which is tau, for example, but it may be acting on something else that's fundamentally important, some other protein that looks like tau, and which is very important in day-to-day function, for example, in the liver, for example, and the drug might be acting on that too. So that's where proteomics comes into play in understanding what else is the drug doing besides what it is supposed to do. Is it doing something harmful or not? And in the clinical trials, I already talked, the number one thing that proteomics we use for in the clinical trials is the biomarkers. We use the biomarkers to select patients, to select how much dose to drug with, and finally to know whether they're responding to the patients are responding to the treatment or not. So within all the omics tools that are available to us in a drug discovery program that is targeted against a protein, I would say proteomics really is the fundamental backbone of how we move forward in drug discovery. This is just reiterating the theme. So one, you might ask, why is there so much protein? Why is there so much focus on the protein? And I'll come to that. Well, one of the things is not all genes produce proteins, right? Certain genes may never produce any proteins. So it's not that simple to go after a gene target. The way to go after a gene target would be to go after a transcription factor, for example. Then you know that there is going to be some protein expression. So proteins really are fundamentally the backbone of drug discovery. Upstream things like RNA and DNA, transcription and translation are slowly becoming important, but to the best of my knowledge, 90+% of the drugs today focus on protein, and that's why proteomics becomes such an important tool. So what makes a protein a target, right? A protein could be an enzyme. A lot of the kinase protein, tyrosine kinase, serine threonine kinases, these are all wonderful targets. Enzymes actually make beautiful targets simply because they have an active site, and using proteomics, you can understand, and X-ray crystal structure, you can understand what that active site is, what are the amino acids involved, and you can design a drug. So enzymes make wonderful protein targets. Structural proteins, sometimes like dystrophin in Duchenne's muscular disease, dystrophin is a structural protein. It's very rare, but sometimes targets can structural protein. Receptor, and we'll talk about SARS-CoV-2, where the whole field will focus on disrupting the ACE2 binding to the spike protein, right? So that's a receptor ligand type of thing, and it's very easy to antagonize. And lastly, as I said, sometimes this is happening more in the inflammation field, a transcription factor that might actually regulate the expression of a certain protein might become a target. So these are typically some of the categories of protein targets that the industry likes to go after. How do we select a target? Now you know what a target is. It's got a protein with all the properties I talked. How do you select the one you want to go after? Ideally, you want to select a target that is the only protein in that. So if you select a kinase target, for example, right? The problem with kinase, although we have been successful, is there are probably 50+ kinases. So first of all, you have to go after the right one and then show that's highly specific for that one. So because to avoid all that, we like to go after a single protein, for example. Tau is a great example. There is no other protein like tau. So going after tau is a no-brainer for that reason. The other, and this is the validation I talked about, it has to be directly associated with causing the disease. It cannot be a phenomenology or it cannot be anecdotal. It has to be directly, which means if I upregulate that, the disease should get worse. If I inhibit the function, the disease should actually get much better. So that's how it is, that you want a target that is directly related. The other thing that you want is that you want your target only to be expressed in the disease organ. And that is done to actually limit the side effects you might get. Again, going back to the tau example, tau is only expressed in the brain. It's never expressed in the muscle or a kidney or there. So if you manipulate tau, it's unlikely that you're going to affect other organs. So expression of your target protein only in the disease organ is a wonderful thing to have. The other interesting thing when we select targets, which is that we like to inhibit the function. That's why I said enzymes make beautiful targets, because there's an active site. I can design a drug that'll go bind to it, inhibit its function. On the other side is trying to increase the activity of an enzyme. It's very difficult. It has to be done through an allosteric mechanism, not through the active site generally. And we don't like that because those are much harder to design. So we prefer to have things to be inhibited, not to upregulate their function, but downregulate. The last thing that, and this is a really nice thing to have, is we like to have protein targets that are actually on the cell surface. So receptor ligands make fantastic targets also for that reason. Why is that? The reason is, in that case, the drug doesn't have to get into the cells or into the organ. You know, the organ, especially for Alzheimer's, there's something called a blood-brain barrier, and it doesn't let drugs to get into the brain at all. So oftentimes you don't have drugs that can get into the cell. But if you have a cell surface target, it's a whole lot easier. So these are some of the simple criteria, and omics, proteomics specifically, comes into hugely into deciding some of these target selection as well. Okay, so how do drugs interact with protein targets? So here's an example I'm showing you with the family of enzymes called tankerases. And first of all, now if the crystal structure of the target is available, it makes our job very much easier because we know what is the active site, and we can then take that active site with all the different amino acids. We'll talk about it more, how it forms the pocket and all that. But virtually in silico, we can actually then select compounds that are already there that might fit into that pocket or not. So upfront itself, we could probably screen 50,000, 100,000 in silico compounds that can fit into the pocket of the active site of the enzyme. And then only work on those. Out of those, maybe only 100 might actually fit very well. So your job is now shrunk down from having to deal with 100,000 compounds to about dealing with 100 compounds. So having a crystal structure is a very important and usually the most useful thing you can have when you have selected your target. Okay, I want to now move on to how proteomics has been applied to a disease that now we are all familiar with, which is SARS-CoV-2 driven COVID. So what are the key features of SARS-CoV-2? On the very left-hand side, you will see everybody has heard of spike protein. So this is the virus, and the way it engages with the host cells, human host cells, is through a protein called spike protein. How does it engage with the host cells? How does it get into the respiratory system? How does it get into the lungs? Is through a receptor called ACE2 that you see. So if you look at the second box, there's the SARS-CoV-2 with the spike protein sticking out, and then you have ACE2 receptor. This is a counter receptor protein that's present on a lot of different cells in the body. The endothelium, the lung is pretty much ubiquitous. So therefore, although the focus of COVID has been on the lung, you will know now that long COVID, which is the effects of COVID that come a few months after the COVID infection, can be affecting the kidney, the heart, because they all have ACE2 there. So the virus can enter all of those organs. So the virus binds to the ACE2 receptor, and as shown on the third box, that's how it gains entry into the cells. And it creates a mayhem from there. As you know, the cytokine storm, the inflammation, and as well as the thrombosis that sets in. So a key feature of SARS-CoV-2 infection is driven by two proteins. One is spike protein on the SARS-CoV-2 virus, and the other is the human ACE2 receptor. So as you can imagine, most of the targets for COVID are actually related to either ACE2 or the spike protein. On the left-hand side, you will see strategies that target ACE2 receptors. Strategy one could be to create soluble forms of the spike protein. Within the spike protein, as you'll see, there's a specific domain called receptor binding domain that engages with ACE2. So one strategy that is being attempted is to design soluble RBD. So this will bind to ACE2, so the actual virus cannot bind. You can also actually make soluble ACE2, for example. So the ACE2 soluble simply means it's floating around. It's not bound to the virus or not bound to any organ. So it's floating around in the blood. It'll see the virus, it'll bind it, as a result of which the virus, by using both these strategies, as is shown on the second figure there, you can see that there, that the virus cannot engage with ACE2. And basically, if the virus cannot enter the human cell, it's dead. There's nothing it can do. Vice versa, you can also have strategies to attack the RBD. And this, which is the domain on the spike protein that's important in binding the virus to ACE2. And RBD has become the #1 target protein or target protein region for COVID-19. Whether it's vaccines, whether it's diagnostics, whether it's new drugs, everybody is focusing on RBD, including the virus. The virus is also focusing in RBD. And when I say that, what it's doing is it's mutating the RBD region to gain superiority, so try and escape from the host defense mechanism. So the whole world of SARS-CoV-2 COVID is actually focusing on this region called RBD, which lies within spike protein. Very interesting portion of that protein, this RBD is, and I'll tell you why. So this is just to show you the structure of spike protein. So spike protein is actually sits on the surface of the virus. It's a part of it is into the virus, but most of it is outside, so that it can bind to go and grab ACE2. As you can see here, the spike protein has actually been broken down into spike protein S1, spike protein S2, spike protein S1. The S1 is really where the RBD, you can see that in the green, RBD lies there. It's on the outside of the cells of the virus particles, and so that it can go and grab onto ACE2. All right. The other parts are, there's also an enzyme too, which is called Tempress. I mean, it's I'm giving you a short form that allows, once the ACE2 is bound to by RBD of the spike protein, then the rest of the ancillary proteins come into play and make sure the virus can successfully get itself into it, right? So RBD is really the star of the movie. If somebody were to make a movie on SARS-CoV-2/COVID, RBD would be the hero, I think. Yeah, so this is just to reiterate that, again, in the orange, you will see the virus, and then you see the spikes coming out. That's a spike protein. And on the human cell, you'll see ACE2. And I just want to reiterate, everything we are doing today to combat SARS-CoV-2/COVID is targeting this protein, whether it is vaccines. All of the vaccine, either using a nucleic acid approach or using a protein subunit approach, the antigen that's going to be used in the vaccines is RBD. There's nothing else. Some people use full spike protein, but a lot of them use RBD. The idea is if you either vaccinate with RBD or produce RBD in human body in small amounts, you start to have neutralizing antibodies that come into play, which will stop the binding of the virus to ACE2. Same with diagnostics. There are many diagnostics now, which is the rapid antigen test or the RAT test you've heard. What are they detecting? They are detecting spike protein. And the drugs. I mean, there are many drugs that some of them we have worked on and published are also focusing on that. So RBD interaction with ACE2 is fundamentally the target for everything that's going on SARS-CoV-2. So why is RBD so important or so interesting? Well, I already said it's important because that's what makes the virus enter into the host cell. Beyond that, the virus is very smart. Once you have developed a vaccine against the original strain, right? What happens is that once you have the original strain, sorry, just one second. Once you have the original strain and you find a vaccine to it, the antibodies to the original strain RBD will actually neutralize it. So the virus has started to mutate itself in the RBD region. Originally we had the alpha, then you have beta, gamma, delta, and now Omicron. The one big change in all of this are that mutations are occurring. So the virus has figured out that if I mutate, change some amino acids in this area, then I might be able to escape the neutralizing antibodies that's being produced by these humans in response to vaccine. So it's trying to constantly outsmart the humans by doing that. So not only does it escape the vaccine when it actually changes or mutates certain amino acids, all of the mutations that have a detrimental effect are in the RBD region. To the best of our knowledge, every single one of them, Delta, Omicron, Gamma, they're all in the RBD region. And some of them make the virus bind tighter to the ACE receptor, as a result of which they can enter, but they're more transmissible. Some of the mutations make them lose the neutralizing activity, and I'll tell you about that too. So just remember, the take home from this slide is RBD mutations are fundamental to how the virus has been moving forward all these times. All right, so one of the things you will see is you may have had the word called escape mutations. There's a big problem. What are escape mutations? Escape mutations are nothing but mutations in the RBD of the virus that actually cause the mutated virus not to be neutralized by existing antibodies. So just to show you in the box here, we created vaccine against the wild type RBD, right? And that we were able to neutralize. The virus mutated itself. It created something called B1.351. When this happened, the neutralizing activity slightly decreased. So delta actually slightly decreased. The neutralizing activity that was generated against the original wild type didn't fit very well with the delta, for example. And so not all the neutralizing could happen, so the virus could escape. Actually, things got even worse. With Omega now, there is about 39 mutations plus in the RBD, and they seem to be escaping all antibodies produced by the vaccines to the wild type. So you can see that having small changes in the RBD, substitution of certain amino acids, gives a huge upper hand to the virus in escaping, and that's why they're called escape mutations, escaping the neutralizing antibodies that we are producing. So sometimes what happens is we are giving ourselves a booster, recently, in the hope that maybe having higher titers of antibodies might stop the Omicron. We don't know how it's going to happen, but Omicron is not being neutralized by most of the vaccine-related antibodies that have been created. So the virus won there. So far, that has won. If with the wild type and delta, we are very happy because the vaccines work pretty much nicely. Omicron, we have started to see some issues. So why is the vaccine success depend on protein antibody interaction? So again, having a crystal structure or structure of these, the RBD domain as well as the antibodies becomes very important. What I'm showing you here in the green is the RBD. And then I'm showing you breaking that, blowing that up a little bit, is that there are different epitopes. Epitopes are just regions of a protein to which antibodies bind. And because of the mutations that have happened, the epitopes are changing. You know, when you mutate a protein, the amino acid change, right? The structure also changes. Inevitable that the structure will change. Once the structure changes, the original epitopes to which you designed your vaccine might be hidden or might be lost or might not no longer be recognizable. So that's why, you know, this interaction between the antibody and the vaccine pretty much dictates the success of your vaccine, right? So if there is too many mutations in the antigen and your antibodies cannot dock very nicely, that vaccine is going to fail. I want to move on and tell you about how omics and protein drug interactions are important in and form the basis of small molecule drugs also. I talked a lot about vaccine, but even in designing small molecule drugs, the interaction of a drug and protein is very important. Here's an example. So this is the structure of a drug. And what is important here is how will the drug fit and bind to the protein in a region of your interest. And usually the interaction between a drug and a protein are either hydrophobic, which means that there are hydrophobic amino acids there, and your drug might have some hydrophobicity, so there'll be an interaction there. The other types of interaction, hydrophilic, there might be charged amino acids on the, for example, an enzyme in the active site, and the drug will have to have charged amino acids and interaction there. So there's hydrogen bonding, there's electrostatic, hydrophilic, hydrophobic. All of these are important in the high affinity binding of a drug to the protein target. So a medicinal chemist, once he has a structure of the amino acid, he will try and tweak the drugs to see if he can fit into that pocket. So not just in vaccines, but the protein structure and proteomics are playing a very fundamental role in us designing drugs as well, to make sure the drugs fits very tightly with the protein target, tau, for example. I also want to tell you that omics are helping us understanding. So one in designing vaccine, the other is designing drugs, selecting target. I mean, all across the value chain of drug discovery, they're playing a very big role. Now, the other big role that omics are playing is trying to better understand the mechanism by which the drug will work. Why do we need to know that? Why do I care about what the mechanism? The reason to understand the mechanism by which a drug works is then you can predict what kind of side effects it might have. So if there is a kinase inhibition is the mechanism, right? We know that there is 50 plus kinases. So it's very possible that your drug might actually bind to more than one kinase and might give you side effects. So knowing the mechanism of action is a good way of predicting the adverse or the side effect profile. And omics have really played. For example, here you can see that this is a Huntington disease cell model shown on the right hand. You throw the compound in, and there are a variety of proteomic technologies you can see to see two sets of experiments can be done. One is what set of genes are being upregulated or downregulated by the compound, right? So that gives you some information. The other is where exactly is it binding and what else is it binding. So there is a lot of exciting things that are coming in, understanding of mechanism of action of drugs since the omics had arrived. There is a saying now that a lot of the drugs that were approved in the 60s and 70s, when we didn't have these beautiful tools, would not have been approved today because they just have too many side effects. Aspirin, as an example, today, if it were to be launched, we know it does so many other things that you don't need it to do, including things like gastrointestinal bleeding and all. So it's only today or in the recent past that we've understood that drugs of the past have many other things to do, and that has only become possible because of our ability to use genomics, proteomics, and metabolomics, I would say. I think it's a very, very safe statement to make. So in summary, I think
Sir, I would like to
omics,
extract.
this is my last slide.
Oh, thank you so much, sir.
Omics have rapidly become the heart of drug discovery. There's no question. Protein targets are the basis of 90+% of the drugs. As you can see, mutations in the protein targets can cause dramatic changes, escape mutations. And proteomics is pretty much useful in target selection, drug selection, mechanisms of action. So long story short, long live omics. Because of omics, I think the likelihood of success of drug discovery is going to dramatically increase. So thank you very much for your time. I don't know if I exceeded by a couple of minutes or so. I don't know. Yeah, I did, but don't kill me for that, please. I'm done.
It was definitely a wonderful talk. We have a lot of questions. I'll mail you out, sir, and then we'll get back to our participants with those questions.
Absolutely, no problem at all. Yeah. All right. Thank you,
so
Mohit.
much,
It was fun being here. Thank you, Sonalika. Yeah.